So let me thank the organizers for having me here. Also, it's been a wonderful meeting. I learned a lot. Also, the talks have been extremely very good quality, and also the posters. Great conference. So what I'm going to do today is to really tell you the kind of work that we do and also get all of your feedback. So most of our lab is the biomedical context that we are interested in is trying to understand how cells age and how does aging-related diseases come about. As all of you know, I don't have to belabor this point. We are the same DNA, trillions of cells, is packed in different ways in different tissues. And the important thing is that aging is inevitable. And with aging, what you have is stiffening of our tissues. So what we are interested in is understanding how this chromatin organization that you all study so much, how does it change during aging? And can we think of ways to push it back through a non-genetic mechanical routes for rejuvenating aging cells? And the other side is what I'll talk today is the subtle changes that happen to the chromatin. Can we capture it well? If we can capture it well, this could be a very ultra-sensitive biomarker for cell states and cell state transitions. So around that, that's the kind of questions that we ask. And today I'm going to talk mostly about how we can bring in AI into this context. So as all of you know, if I take a piece of tissue, every nuclei has very distinct shapes. It's not round, right? I mean, this is a picture of a tissue. And some cells, some nuclei are round, some are very stretched. The reason is that the extracellular microenvironment that cells see influences the entire cellular architecture, nuclear architecture, and the chromatin architecture. So what we study at the scale of gene transcription, condensates, and so on, and chromosome organization, all of this is intimately linked with the extracellular microenvironment through a number of biochemical and biophysical linkages that come about. And therefore, the way that the chromatin is organized is a functional integrator of what cells see on the outside and what cells produce in terms of the output. And so because there is such a tight link between the physical space of the nucleus and the cells, we have argued that there is a cell type specific optimization, that if I look at the physical space of the cell and the nucleus, I should be able to think of the biochemical, complex, multidimensional biochemistry that exists in the cell. So what I'll try and belabor you today is how we can bring in machine learning to prove such linkages exist, such cell type specific optimization comes about. And if this optimization is lost, what you have is loss of homeostasis, and you all study this quite a bit more than we do, that the nuclei, for example, in aging or premature aging, neurodegeneration, in cancer, most of this chromatin organization gets altered. So can we think of imaging these alterations and see if we can learn from those images using deep learning techniques? So diseases, as you know, doesn't happen overnight. It's a progressive process. As a function of aging, this is, for example, tumors in the body, different types of tumors. As you see, as we age, depending on the time scale, depending on the tissue of origin, cell state transitions occur, and eventually these give rise to altered cell states and diseased cell states. So what we thought early on, because of the success that came about by thinking of images as hierarchical structure, hierarchical future representation, can we implement such interesting ideas that came about? In fact, this really reinvented the whole area of deep learning. Can we use this kind of a representation and bring it to learn about nuclei, cells, architecture, tissues, and so on? So that's what we did. Very early on, we jumped on this together with my friend and colleague, Caroline Wooler's lab. Our labs work together. Since, as all of you know, which I don't need to say this again, every nuclei has very precisely organized chromatin structure. Can we take pictures, tons of pictures, thousands of pictures of those nuclei in its native context, wherever they are, right? Or in culture, or if they're differentiating, or they're undergoing disease progression. We take a lot of pictures of the nuclei in intact context, functional context, and learn these images. Are there hierarchical structural features in these images that we can learn? And we do that both by unsupervised learning or supervised learning. And what we get then is every nuclei, then we can have high dimensional information on how the DNA is packed at different scales. And this depends on the microscope that you use. If you use a super resolution microscope, you can extract much more information. If you use a confocal or a wide field microscope, you get a little less, but quite a bit, so that you can use that information to group your cells, right? So this is like taking a single cell RNA-seq now, right? If I take a single cell RNA-seq, what I have is a high dimensional information of the protein expression or the RNA expression. In the same manner, for every nuclei, I have a high dimensional information of the folds of the chromatin at different scales. And this turns out to be extremely useful in thinking about cell states and cell state transitions. So what we do is we learn a large number of features, as I said, and we can actually compartmentalize that to nuclear morphometric features, tons of these features, or chromatin compaction features at different scales, or heterochromatin organizational features at different scales. And so now we have hundreds of features by just taking a picture of one nuclei, and we take thousands of pictures of the nuclei so that we have very good statistical representation of a cell state. So with this, we can do quite a bit. Simple example that we tried very early on is just plate different types of cancer cells, metastatic, benign, invasive cancer cell lines, take pictures of them, learn these pictures, and ask, can I identify which group of cells are metastatic, which group of cells are benign, which group of cells are invasive cancer cells? So these are reduced dimensionality plots, basically because every nuclei, as I said, has many features. Now we can reduce the dimension and ask, this cohort of cells have features that they share similar features. In addition, as I said, diseases happen over time. It doesn't happen overnight. So how do we think of tracing lineages, single cell lineages, in an ensemble of tissue context where disease is progressing very slowly? So we now developed methods that you can take pictures of the disease states at different time points. Let's say healthy individuals, normal state, at a little intermediate stage of the disease, late stage, and so on and so forth. But because we have pictures of chromatin in different stages, we can then use a method that we have implemented here, an optimal transport method, which basically does take these images into a latent space where basically these images have some distribution because every cell had some chromatin state. These chromatin states are some distribution amongst a group of cells. Then we go into this latent space, learn how to transport them from one state to the other. And happy to talk to you offline about some of the methods. If we can do that, what we can then construct is an envelope of how every cell's chromatin structure has changed during the disease progression. So this is what we do. This has been extremely successful. I'll show you some examples of how we can actually trace lineages using such methods. The other thing we want to do, which we have spent quite a bit of time now, is all of you are very experts in many data domains. As all of you know, cells come with both physical space, also complex biochemical space. Can we go from one data domain to the other data domain? Because some data is easy to access, some data is harder to access. For example, if I want to do single cell sequencing of all the cells in the tissue, it's a bit daunting task. Or if I want to do ATAC-seq of all the cells in my tissue at single cell resolution, still a challenging task right now. Or if I want to do Hi-C, and so on and so forth. So our idea was that, can we learn what exists in these spaces in terms of the data? For example, you know that if I take a single cell RNA-seq, you know that which proteins are expressed, which RNA is there in that cell. Or if I take accessibility or chromosome contact maps, I know which contacts are persisting. But can we take images, and from the images can we go into these domains? Because then if you give me a picture, I can tell you what that cell will produce in terms of the expression, right? So there's a new method that we introduced. This is one of the methods that really started the field as well, and now there's a lot of efforts in really thinking about how do we go from different data domains. This is a work that I closely do with Caroline's group. So the idea is very, in terms of the technique, it's very complicated, but in terms of the idea, it's very simple, that let's learn what is there for the cell in terms of the single cell sequencing, all the dimensions that exist. Let's learn what exists in the imaging space. What are the various features that exist in the way DNA is folded? Let's use autoencoder. These are encoders that can actually bring data into representing data in ways that you can actually transport between one to the other. So we take these different data sets, go into what is called an integrated latent space, and ask, can I learn how to move these data into this domain? If I know how to learn that, then what I get out from a picture is already a sequencing information. Or can I learn how to think about what contacts that these cells would have, chromosome contacts? It depends on the imaging resolution that you've taken. You can then convert these images into a chromosome contact map or an ATAC-seq. And more recently now, some of these papers are out already and some of them are coming out. We can also think about transcription regulatory networks. How do we, because understanding transcription regulatory networks is a daunting task, as some of you know. Can we go convert from an imaging space into learning gene regulatory networks? Or can we learn where proteins are localized in the cell? For example, in the cytoplasm or the nucleus, depending on the transcription factor, cofactors, depending on the spatial localization, they turn on different gene regulatory programs. So we now have methods now where we can actually link the genome organization with proteins where they're sitting in the cell, and we can then think about the dynamics of these processes. And also we can actually go to other spaces of the cell now. So with this, I'll just show you one or two applications. One application is to think about, let's take real tissue biopsies of a human brain or a mouse brain. The first work was a mouse brain, and the next work that's coming out is in the human postmortem brain. The question we asked is, during neurodegeneration, can we think about looking at the chromatin state of every cell in the tissue and ask which regions are poised for neurodegeneration, where the amyloid deposits might come about? Also, can we differentiate between frontal dementia versus Parkinson's versus Alzheimer's? So we are partnered with the UK Brain Bank, where they have some of the best postmortem slices preserved. And we can now directly predict, and also we combine that with spatial transcriptomics to see what is the power of chromatin organization with the spatial expression maps that we have on the same slices. And that gives you a very unique possibility to look at imaging space of the chromatin and think about understanding diseases. The other context that we work on quite a bit is, can we look at the envelope of the breast tumor progression in human biopsies? So these are tissue microarrays of different stages of the tumor, as you see. And these are images. Every dot is a nuclei in that tissue biopsy. And now we learn every nuclei's chromatin state in these tissue biopsies and put a score. For want of reason, we call this a mechanogenomic score because it couples extracellular microenvironment with the chromatin state. And each nuclei then gets a score. And now you can actually see that once you follow this tissue biopsies of different stages, we can literally say which cell is switching from a normal state to a disease state. And this is very hard to do at this time because if I want to understand the disease progression in tissue biopsies, I really need to do transcriptomics, or I collect the cells and do transcriptomes of every cell, or I do spatial transcriptomics of these tissue biopsies, which is also extremely difficult. So now by just learning the features of the chromatin state, we can actually start saying which cell is going to switch its state from its healthy to a disease state, and what is the spatial reorganization that happens in these tissues. So as an example, we now pushed this quite a bit. So by looking at all the nuclei in the tissue biopsy, there are thousands of nuclei, right? More than 400,000 or something. We collect all the nuclei, or it depends on the size of your biopsy, and ask and use interesting clustering methods to ask, can we cluster this nuclei based on the chromatin state? And what we can actually now do is cluster them into many, many clusters. Basically, each cluster shares some similarity between the nuclear state, right? Now we can go back and ask which cluster belongs to healthy state and which cluster belongs to the different states of the disease, and annotate them and compare with the pathology as well. What came out as a surprise is that when you looked at the healthy cell state, healthy tissues, tissue biopsies from the breast, human breast tumor, breast patients, breast donors, you have residual nuclear state, chromatin state already present in the healthy tissues. And so you enrich for a lot of clusters with chromatin state which are more similar to healthy state. But you also have clusters, the typical chromatin state, which are already in a intermediate disease or late stage disease state, but in very, very small proportions. Right? This is the cell state proportion. This is a cartoon saying that this completely disease state, we already start seeing the chromatin state of the disease state in the healthy state. That means that we now think that tumor progression with aging is really reorganizing these cell states in a manner that they can then, if they become proliferative, some of the state, then they can actually go on to divide rapidly, right? And then you can then go on to have metastasis like this. And so there is a big challenge, unmet medical challenge that we're looking at, which is when you have ductal breast cancer, the current treatment option in the world today is either surgery. It's a very early stage though, right? It's a very early stage, the ductal carcinoma in situ. But because of the lack of knowledge pathologically, which one go on to become invasive, the treatment options are either removal of the breast or removal of the local tissue by surgery or radiation, chemo, whatever you have, right? But epidemiologically, over the last 50 years, what's become clear is that when you have this DCIS, ductal carcinoma, only 30% of the patients go on to become invasive, become cancerous, right? But there's no way to really know which one becomes invasive, which one doesn't become invasive. And so there's a big unmet challenge that we're looking at. Can we look at these chromatin states in these healthy DCIS tissue biopsies? Can we predict which of them will become invasive, which of them will not become invasive? And we are still not there, but we know how to really trace the tumor progression. We still don't know which one to predict, because it's also, in fact, when this paper came out last month, many patients have written to me, I'm diagnosed with this stage. Can you tell us what I should do? And we don't have an answer still, but that's something that we are working at. So all of these things, trying to diagnose disease, what I talked so far, requires invasive approaches. Somehow the patient has observed a disease, so you go to a clinic in a mammogram or something like that, you have to have an invasive biopsy, and the pathologist decides what stage of the disease you have. So we are now pushing for whether we can, not just us, the whole world is pushing for early diagnostics. And one way to push for early diagnostics is non-invasive methods, because as many of you know, when we have a disease in the body, they secrete signals, and these signals eventually land up in the bloodstream. These signals could be exosomes, ctDNA from the cancer cells or any other disease cells itself, or cancer cells themselves, circulating tumor cells, or secretome signals. So there are many signals in the blood as the disease grows, whether it's neurodegeneration, whether it's metabolic disorders, whether it's cancer, whatever you have. So we wanted to think of an orthogonal idea, because one of the cells are immune cells, so the most sensitive cells in our body, as you know, because they react. When they see a ligand, when they see some signals, they react because they're to sort of start an immune response. So we wondered that if you take blood cells, white blood cells or peripheral mononuclear blood cells, take pictures of image the white blood cell chromatin structure and ask, just by those pictures, can you say there is a disease in the body or not, right? We've had a lot of luck with this. We take white blood cells, purify them from real human patients, and there are many methods to purify them. Put on a good microscope, take a lot of pictures of the chromatin states, and use our machine deep learning methods to ask which of those cells coming from which patient are cancerous or not, right? And we can actually annotate, the state of the cancer. So this is very powerful for really enabling early cancer detection or early disease diagnostics. But also there's an unmet challenge about therapeutic interventions, because when we go to the clinic, we get a therapy, but we don't know whether it worked or not, because there's no way, at least until there is a relapse of the disease in the field now, right? So now we are actually pushing this for evaluating therapies. One example is an ongoing trial is about proton therapy, because proton therapy is a form of therapy, an advanced form of tumor therapy or for any other disease, which uses protons as radiation source. And this is an example of patients come to the clinic. We take blood from the patients prior therapy. During therapy, we take blood samples, and end of therapy, we take blood samples and a follow-up, 3 months and 6 months, and we learn chromatin states, use deep learning to learn chromatin states in this and ask, can we start thinking about assigning how the blood cell chromatin states change with therapy? And this is now a finishing trial now. We're going to put out the data soon. It's a 150 patient trial with 5 time points of the therapy, proton therapy. And what the clinicians are already excited is, when they provide proton therapy, not all patients respond. Only some of them respond to therapy, and many of them don't respond. So now they're excited that they can now, during the therapy process, because there's no way that they can assign this, they can then correct their therapy model to sort of better implement these therapeutic things. So this has led to a number of ongoing clinical trials in our group, one for early cancer detection. We have a human trial that's finishing for metabolic disorders. We can actually detect from the blood chromatin states onset of metabolic disorders and progression. And we have a clinical trial starting on neurodegeneration, whether we can pick up neurodegenerative signals in the blood cell chromatin state. And whether we can actually think of efficacy of therapies. What I showed you here is proton therapy, but we are now working with clinicians for surgery, chemotherapy, radiation therapy of all kinds. And can we use this as a generalized model for doing this? But to do that, one of the important things we need is really having a good data for healthy donors. If we know how the chromatin states of our blood cells look in healthy donors, then we know what the outliers are. So this is a global public health project that we have started in a group. It's primarily Swiss-based right now because it turned out to be too. I was also thinking of doing it in India as well, and other places it turned out to administratively too complicated. So we now started this, and we have now a number of foundations that are very keen to fund us in large proportions. The idea is simple. It's an open source public health project to create a chromatin imaging atlas of our blood cells in different age groups and different sex groups. And how does the chromatin look like in all these healthy individuals? So we partnered with the Swiss Red Cross. They're excited because they have a good gold standard in collecting blood because they implant the blood. So we work with them, and we are trying to generate a huge cohort of blood cell chromatin states in healthy people. And once we have this, then we can actually start looking at outliers, because when a person comes to your clinic, if you have this reference atlas, then you can compare with the reference atlas your blood cell state. And to do that, we are trying to push miniaturized microfluidic devices and miniaturized cameras, optics, so that you can take pictures of the blood cells easily. Because you can't take a confocal microscope to a small clinic in Africa or in various places. So we're now perfecting this technique of taking blood with a microfluidic device, using your own mobile phone to take pictures. We have improved the optics that we can actually get very high resolution images of the blood cells, of the cells, that can actually give you enough information to learn from each of these nuclei, high dimension information of how our chromatin is organized in these blood cells. So happy to talk to you about this public health project. I'm very passionate about it. We'll also come to India at some point. All of your help would be needed. But for now, we will focus entirely in Switzerland, which is easy to do for me. And then we have a lot of foundations that are interested. So to summarize, what I told you today is a simple idea that cells in our extracellular microenvironment sends both physical and biochemical signals and relay those signals to alter chromatin states. And as I said, the chromatin state is alterations in them is intimately linked to the functional space, as all of you know. And we've developed state-of-the-art machine learning techniques to be able to learn this integration between this space and the biochemical space. And we think that learning these two spaces, we have new opportunities to translate this information, the simple, cost-effective chromatin imaging states, to really not only clinical diagnostics, but start understanding molecular mechanisms. Also, we're now using other types of labels on the chromatin, whatever labels we have, post-translational modifications, POL2, all kinds of transcription factors that you all work with, and nuclear bodies, and so on and so forth. If we can learn all of them, it's a huge information that we can generate to think about functional function. Also, we are now pushing techniques to do live cell tracking and learn the live cell tracking pictures, because as all of you know, if systems are ergodic, I can take a lot of pictures, static pictures, or I can take a few cells, and in time I can take them, the time series. Then both are sort of give you similar information. So we're now trying to push for recording this in time so that we can actually see how biochemical network space is changing as the cells are either differentiating. We work on reprogramming, so we're looking at reprogramming events and rejuvenation events as cells age. So with that, let me thank a whole group of talented colleagues. We're always recruiting a lot of funding from various sources. ETH created a professorship for me, so we do have resources. and I'm also an affiliate member of the Broad Institute. Caroline Wooler's group at MIT and Broad and our group. It's a joint group. We work together, funded by other sources. Thank you so much.
Thank you, Shivashankar. We'll take 2 quick questions for interest of time.
Well, I'm not sure this is a quick question, but why just chromatin? I mean, if you were just to do classification of whole images, I guess how much would the nucleus contribute as a feature into the classifier?
So you can use cytoskeleton, you can use extracellular matrix, you can use any parts of the tissue to learn. The most stable state in a given cell, in a given tissue, is the chromatin state. And so we've not tried all other things, but the robust signals are at the level of chromatin, and that's why we...
Just one question. So you talked about optimal transport.
Where are you?
Here.
Ah, you're there. Okay.
So you talked about, you just had a slide, so optimal transport. How are you using it? In what context?
So let's say you have a group of pictures. You have a picture of a state of cells. And so what that gives you is a distribution of all the states of the chromatin that exists in that cohort, right? And now I take a picture of another state of the cell, let's say normal state and metastatic state of the tissue. And I create a distribution of all the ways that the chromatin is organized. And then maybe I take an intermediate state when the disease is progressing in some intermediate state. Now I have a distribution. And what we're trying to do is to transport information in this distribution from one distribution to the other, learn how to transport it. And these are methods that have been invented and developed in statistics, and now we can actually borrow those methods and start seeing what is a way that we can actually translate one distribution to the other distribution to the other. And if we learn how to do it, then we learn how to transport each cell's feature, whatever it is. Here it's chromatin, but any other features. What that creates you then is creating a lineage of envelope of how the cell states change, although you didn't have data in between, right? Because I didn't have data in between.
I'm just curious about the cost function. I'm aware of the method, but I'm just interested in how do you actually define a cost function? Because that's...
We can talk offline. It's a bit technical in terms of the cost function. You need to optimize. So it's primarily now it's not a black box. It's an art, because transporting each distribution may not be just simple Gaussian distributions. It could be log normal distributions. It could be any other kinds of distribution. So each state, we need to learn how to transport those distributions. What is the cost function that is needed? The cost function may not be the same for each of the transport.
I mean, the reason I'm asking is because we're actually thinking about that for a different problem in biology. But again, the issue is like the cost function. How do you define that?
How do you optimize that cost function is actually very important.
Thanks a lot.
And it's very system specific. Yeah.
Can I?
One more, yes,
Very short question. Okay. So this might be related to the previous questions to some extent, especially in the context of tumor cells and the cross sectional of tumor cells and all the cell types that are there, which are continuously evolving. As one progresses from. I was wondering, you might have tissue sections from different stages, perhaps. So the spatial distribution of cells have also changed as you have moved ahead across time. Mm-hmm. Now, how does one bring that back? Because if you're looking at a bunch of different cell types and a bunch of different cell types having different conformational states in terms of chromatin states, and these are across time evolving, how does one sort of put that all together in a scenario, or you let the system learn from all the stochasticity that is already built into the system?
The tissue reorganization is fundamental to changes in either in development or in disease, right? So there's dynamic changes that's happening. So now what you have is pictures of every cell or nuclei in that tissue. And we learn their spatial locations as well. Neighborhood, spatial locations, all kinds of things that we can actually learn using deep learning techniques. Now, if we have that in different states, we already learned where these things move around. As I told you for the DCIS, this is actually by far the largest data set in the world now, what we have created, of about it's not as large, it's about 650 patient samples of just the DCIS, transitioning to invasive ductal carcinoma. What you can then do is learn how the dynamic spatial reorganization happens, because we can now fingerprint every cell state using how the chromatin is organized. And we learn the neighborhoods of these cell states across the entire tissue, right? Not just one or two neighbors. Once you have that picture, you can actually now start learning, if you had data for the disease progression, you can start learning how the spatial reorganization of the tissue happens, right? And now we are applying this for aging context. As we age, in healthy aging, how does the tissue architecture change, and how can we when the disease onset happens, can we bring in interventions early on?
Brilliant. Thanks, Shiva. Very exciting work.
Thank you. So that marks the last talk of the session, as well as the last talk of the conference. Let's thank all the speakers for sharing their research with us, the organizers for the terrific organization, and the attendees for your active participation. Thank you all.
Thank you, Nishan.